Good afternoon, all of you. It's a great pleasure to be here. Thanks to Dr. Deepa and Dr. Kavita for inviting me to give a talk here. We work on human genetics, particularly understanding complex history of Indian populations, and also some of the disease which are highly prevalent among Indians. So my talk basically to cover both population as well as the medical genetics. And now it's everybody can understand, even after sequencing of the whole genome of both human and many other organisms, that we, the human, have diverged from our close relative in terms of the evolution, chimpanzee, for about 5 to 7 million years ago. And during this period, there's a lot of anatomical changes have taken place. Particularly the brain size keep increasing. In case of Australopithecus, was only 35% of the modern human size-wise, then increased to 50% in Homo habilis, then Homo ergaster has about 60% of the modern human, and in Neanderthal it was predicted that the brain size was even bigger than the human brain size, but I don't know how or why, what was the reason the Neanderthal become extinct. And one of the well-known anthropologists and archaeologists from Cambridge, Paul Millers, so he wrote in one of his articles that the violent behavior of human is responsible for vanishing of Neanderthals. In addition to that, the modern human and from last 5 to 7 million years, different species have learned how to make the tool for killing animal and many other purpose. So coming to modern human and its origin, it was again well established fact that the modern human originated in Africa about 160,000 years back. And the question is, how did they spread from Africa to other continents? And there are two hypotheses. One is the northern land route, where a group of historians, archaeologists, anthropologists, including linguists, believed that the first modern human must have taken the northern land route. But the other group, they don't believe that, because the climatic condition is not favoring or must have favored to take northern land route. So the other group believed that the modern human must have taken the southern coastal route through the Indian coast. And based on the archaeological evidence, it was established that the modern human lived in Australia about 50,000 years back, in China about 39,000 years back, in Middle East 47,000 years back, and in Europe 40,000 years back. So unfortunately, we don't have good archaeological evidence from India because of the climatic conditions. Very difficult to predict when did the modern human lived or arrived in Indian subcontinent. But however, although we don't have the archaeological evidence, but we have large number of human population exists in India who preserve their genetic material very intact. Therefore, that would we thought that it will be useful for studying the genetic information of modern human to understand the Indian population history, or when did the modern human arrived in India, from there where did they go, and many other complex issues, including the medical issues. And to let you know that in India there are 4,635 anthropologically well defined populations. That's a very, very large. Anywhere in the world you don't see such a high human diversity. that includes several castes, several tribes, and primitive tribes, including hunters and gatherers. There are 36 groups who just hunt for their food. They don't do agriculture or they don't do any other job. And these populations, 4,635 populations, are well stratified in two broad ways. One is the social stratifications, like caste and non-caste, and again non-caste we have tribes and religious group. Again, every group has subgroup, breeding isolates and exogamous unit, and every single population maintain their endogamy, and maintaining their endogamy for the last several thousands of years. Therefore, whatever the genetic change happened in this population is exclusively going to be present here. You may not find that here. So that helps in identifying every single population. So in addition to the social stratification like caste and tribal groups, another major classification is the linguistic classification. There are four major language families in India. One is the Dravidian languages. All the four southern states, people speak Dravidian language. Here in the state, Kannada, Tamil Nadu, Tamil, Kerala, Malayalam, and in Andhra Pradesh, they speak Telugu. Of course, now Andhra Pradesh has become two, Simandra and Telangana. So anyway, both will speak in Telugu. So now the 5 states speak Dravidian language. And this is considered to be one of the oldest language. Even the people are the oldest people, which we'll see in coming slide. The second one is Austroasiatic. People live in this particular area, they speak Austroasiatic language, and they are also considered to be one of the oldest settlers in India, but they are not that old as the Dravidians. The third category is Tibeto-Burman. People from northeast and northern part of India, they speak Tibeto-Burman language, and majority of the Indians speak the Indo-European language. About 80% speaks Indo-European language. The social stratification and the linguistic classification, so they act as a barrier for gene flow between the population group. And since every single population been following a strict endogamy marriage practice, as I mentioned earlier, so every single population can be identified very distinctly. So in addition to the 4 major language families, we have group of islands where there are 4 tribal groups who typically look like Africans, and 2 population group who look like Southeast or East Asian population. So these groups of language has been considered as a Andamanese language. So in broad sense, there are 4 plus this language, 5 major language has been classified. So in addition to the indigenous population, what we have been discussed, and there are several populations who have migrated due to many reasons like invasion, and these are this population have migrated very recently. So we have 4,600 population plus some of these group makes the Indian population structure much more complex than any other population group. So one can say that India is an ethnic museum because we have large number of population groups. And we have been working on different populations for last at least 15 years to understand the complexity and how these many groups have evolved and many other areas. While studying, we were thrilled to know that the first out of Africa human is in Indian subcontinent. They are none other than the population group which I mentioned called Andamanese. Okay, so this is their typical living habit. So there are 572 islands, very small group of islands. The 4 population which I mentioned who typically look like Africans, Java, Great Andamanese, Sentinelese, Onge, and these 2 groups, Nicobaris and Champagne, they typically look like East Asian population, as you see here. These are the Andamanis, and these are Nicobaris and Shetland. And their number is very, very small. You see that they are even less than 20, and these are the group vanished, and very, very small number of individuals are there. Even Bo, the last lady who died very recently from Bo group, only Jeru is surviving now. And this number, as you see, they are very, very small. Of course, one issue could be because it's a highly endogamous group. They are living in isolation for several thousands of years. I'll tell you how many thousand years. That's one reason, highly endogamous group. But still, they have lot more variations. The other reason why their number is very less is when Britishers captured India, they wanted to construct the cellular jail to put all the freedom fighters there. So they constructed the cellular jail, and some of you, if you had a chance, I don't know, one can see it's very, very, what you say, that the arrangement of the rooms in that is very, no, very unique. One cannot see who is there in the other room. Such kind of, that the cellular jail was constructed. So that time they killed many of these tribal groups. And I don't know what was the reason. They took the skull and the air sample and kept it in the museum, the Cambridge Museum. Now, of course, that was available for us to analyze some of the air samples and the skull to compare the present day modern human DNA. So that could be one of the reasons for their number is very, very small. So we collected the sample from 3 groups. Of course, we could not collect from centenaries. Nobody could go near them. They are in different islands. If somebody goes there, they will shoot with arrow. Okay, so that's the end of the. So nobody made any attempt to go there. So we collected from these groups and the Nicobaris. And initially, as all of you are aware, the mitochondrial DNA is the right marker to trace the maternal lineage because of its uniparental inheritance and many other uniqueness of this. So initially, we sequence the hypervariable region because that's where we find much more variations. Then compare that with the different population, and to our surprise, the hypervariable region, typically it was looking like the South Asian group. It was not unique, it was not similar to Africans, although the phenotypic picture is like Africans. And it was kind of bothering us to see how the typical feature of Africans can have the mitochondrial hypervariable sequence of South Asia. So therefore, we went on to sequence the complete mitochondrial DNA, whether that would address any issue. So based on the mutation exists in the mitochondrial DNA, one can assign every individual into the mitochondrial haplogroup. So wherever, so these are the L-derived haplogroup typically found in Africa. And if in addition to these mutations, somebody has additional mutations, they will fall into M. Majority of the Indians are with the M haplogroup. Same way, as the mutation increases, then they will fall into different haplogroup. And you can see the different continent has different haplogroup which is predominant in those areas. So we thought that once we sequence, we could assign the haplogroup. Particularly, we thought that they may fall in L-derived haplogroup. But to our surprise, so they are not falling in any of the known haplogroup because they acquired unique mutation because they are in isolation for several thousands of years. Right? So they acquired unique mutation. Therefore, we assigned a new haplogroup called as M31 and M32. Okay, these mutations are not present in any population in the world. Okay, so we assigned a new haplogroup, and one can also calculate the age of the population by looking at the number of mutations. Different mutation rate has a different region of the mitochondrial DNA. Like for example, if you take hypervariable regions, then the mutation rate is very high. The coding region, the mutation rate is very very low. So based on that, one can estimate the age of every population. So in this case, we estimated that they are the first modern human migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 60 to 65,000 years back. So the migration is the first wave of migration out of Africa, and it happened about 60 to 65,000 years back. So this was satisfied, and we don't know until we publish on the same issue. Macaulay and their colleagues have analyzed. There's a population called Orang Asli in Malaysia, and there are again aboriginal population, and they also come to the conclusion that the first wave of migration must have taken place through southern coastal route. So this has given again additional evidence that the first wave of migration taken place through coast, and Andaman the first modern human migrated out of Africa, and the previous study suggests that they might have further moved towards southeast, then reached Australia. So Australian aboriginals also typically look like these populations. So having the mitochondrial information for about 15,000 individuals from several population groups, same way the Y chromosome information again for many other groups, we thought that now it's time for look at the autosomal information which inherits from both the parents. So we carefully selected population of the individual from all the language families like Dravidian, Indo-European, Austroasiatic, Tibeto-Burman, and Andamanese. We also took care of taking different social groups, including upper caste, middle caste, lower caste, and the tribal groups. And again, based on the Y chromosome and mitochondrial information, so we wanted to keep as diverse as possible and cover most of the Indian population. And we also used HapMap and human genome diversity panel data to compare our data. So using these, we did PCA, principal component analysis, where all these are Indian groups. We have Chinese HapMap sample, Eurubans is an African HapMap sample, and European HapMap sample. Now this slide alone can tell the complexity of Indian population. You see that every population here forming a tight cluster. There are many individuals, but you see here that every single Indian population are forming a cluster. So that tells that the India is not inhabited by single large population. One can conclude that India is inhabited by large number of small population. So this cluster, individual cluster, is because the genetic difference between these two groups and these two groups. So every single population you take, they are different, but again they are not totally different as we see here, African and European and the Chinese, but again there is some kind of genetic affinities with each population. There's no distinct, no, either you can say north or south or east or west. Okay, there are differences, but still there is a connection. Then what we predicted was that in the prehistoric India, the two ancestral populations, one we call them as ancestral South Indian, other one is ancestral North Indian. And the ancestral South Indians, they show affinities or genetic relationship with North people from North, as we have seen in the previous PCA plot. They also show genetic affinities with the Andamanis. So that means while Andamanis were migrating, some group has entered into southern part of India, and they gave rise to many population. During the process, the South Indians, we lost the typical original features of Andamanis, but still we have genetic relationship, not 100%, but there's some genetic affinities. On the other hand, and they don't have any genetic affinities outside India. Okay, that's another major issue. And the ancient North Indians, probably they must have arrived much later, 35 to 40,000 years back, and they show different group. If you take the tribal group of tribal group from North India, still they have 30% genetic affinities with Europeans. The highest genetic affinities is the Kashmir Pandits. They have up to 70% genetic affinities with Europeans. Okay, so the ancient North Indians show up to 70% genetic affinities with Europeans. The ancient South Indian, they show affinities with the Andamanis, but they don't show genetic affinities with outside India. So what we predicted was that if they are two distinct, ASI and ANI, how did they show this kind of affinities again? So we thought that there could be admixture between these two groups sometime later after they establish their presence here. So that we could not establish at the time. Then we thought that it would be worthwhile to establish when did the admixture took place between these two groups. Then we took additional population, 72, 73 populations, using previous information and the additional sample from south and the north. Then we estimated that the ASI, as I mentioned, gave rise to many populations. ANI gave rise to many populations. And some point of time they admixed, and now we predicted that that admixture must have took place between last 4,000 to 2,000 years back. So that's the reason, although they are two distinct groups, then still we find genetic affinities between the group. So after the admixture, that means after 2,000 years back, every population again started maintaining the endogamy marriage practice. They were two distinct, the admixture, then everybody, every single population maintaining their endogamy marriage practice. That's why we find difference between this and this and this. So, and this is a time probably the caste system also must have been introduced here. But the population difference and the following endogamy marriage practice is much older than the caste system in India. So we looked at the population, how or when did the population arrive in India, what is the relationship. But now, what is the complexity in terms of the health issue? Okay, are we also getting or having the same mutation as the people from rest of the world has, or something different? So to address that, we have looked at the FST first, population specific difference. We calculated among Indians is 0.01, higher than Europeans. Hope that number is here in this audience, and so that was lesser than what we found in India. So that shows that the difference between the population is much higher in India. So that can be explained by the founder events. You know that most of the Indian populations are small in number. That could happen like one family, one parent, 3 or 4 children, they must have migrated or diverged from a larger group, settled somewhere. After a few generations, the 4 children again, they have 3, 4 children, they have again 3, 4 children in the next generation. Maybe in 10 generations, they make one village. Probably they make one, must have named some population or a tribal name. Okay. Since they all evolved from a single parent, originated from a single parent, and marrying within the group, they are genetically very, very unique. Okay, so that's the reason we still find the population specific marker or disease. So what we predicted, because of this, there could be many recessive disease, many population specific disease, and there are some mutations which are Indian specific. So I am going to just give you one example for this. Let's look at the population specific disease condition. Is not really disease, but medically very important. It's a population called Vysya. They are in Andhra Pradesh, very small group. Although we don't have a typical marker to identify the reason for pseudocholinase enzyme deficiency, but it's medically very important. The Vysya group, if they take anesthesia, most often they may not come out of the anesthesia. Sometimes it may lead to paralysis, okay, because of the pseudocholinase enzyme deficiency. Even today, somebody goes for major surgery, the doctors or anesthetists, they ask, are you belongs to Vysya or not? If they belongs to Vysya, they don't give the regular anesthesia. They give different combinations of anesthesia. Although we don't have marker, but what we predicted is the last 108 generation, they are maintaining their endogamy marriage practice. There's no gene flow from outside, or even nobody has gone from Vysya community outside to marry someone. So because of that, it's not spreading to the other populations, and also not diluting because of the gene flow inflow of the normal condition. It's not diluting in this particular population. This is just one example for population. There are many things in Karnataka. There's a place called Handikudu, Shimoga district. There, majority of the individuals are very short with bone abnormality, and the name of the disease itself is now called as Handikudu syndrome. The name of the place itself, and that was typically found only in that region, nowhere else. So that's an example for population specific disease or health condition, and this is because high endogamy marriage practice. The second example is the regional specific disease. There are some disease which are present only in particular region. There's a disease called Madras motor neuron disease. This is one of the neuromuscular disease, and the phenotypes are typically different from other neuromuscular disease. And this was found for the first time in Chennai. Earlier used to call as Madras. Okay, that's why it's named after Madras motor neuron disease. But later, this was also found in nearby cities like Chittoor and Tirupati. Okay, they are very close by cities. The reason could be some individual or family members must have migrated from there and settled in Chittoor and Tirupati. Therefore, we are saying that in a radius of 200-300 kilometers. The third category, what I was telling, is that there are some mutations which are Indian specific. There's a disease called myosin binding proteins. There's a gene called myosin binding protein C3. If there is a mutation in this gene, it causes cardiomyopathy. But we found there's a mutation of 25 base pair deletion. It's in the intron, not even the exon. Okay, that skips away one of the wild type exon. Therefore, there's a truncation of the protein and leads to cardiomyopathy. There are three different kind of cardiomyopathy. One is a dilated cardiomyopathy, where the ventricle enlarges beyond the normal size. Other one is hypertrophic cardiomyopathy, where the heart muscle becomes thick and the intraventricular septum, as you see, is very thick. The third category is restrictive cardiomyopathy, where the heart muscle will be much more stiff than the normal individual. So all these conditions, at some point of time, their age suddenly stop functioning, unable to pump the blood. Therefore, this is sudden cardiac arrest and the individual die. But in this case, the 25 base pair deletion, if it is in homozygous condition, 2 copies of the chromosome carry this mutation, the individual die very early age, less than 10 years. So that we found after analyzing the postmortem tissue. We found 3 children less than 5 years old carry the homozygous deletion. Whereas if it's in the heterozygous condition, 1 chromosome with the mutation, 1 with the normal, then the individual survive up to 40 or 45 years without any symptoms. Suddenly they collapse while walking or playing, right, without any symptom. You might have heard that somebody while walking died, right? So it leads to very, very very serious problem. When we looked at the histopathology, the cardiomyocytes of the individuals who carry the mutations are disarrayed. It's not like a normal tissue. So that again we did some in vitro study where we used neonatal rat cardiomyocytes. Then we cloned both wild type and mutant, overexpressed on the neonatal cardiomyocytes. And this is a wild type overexpression. You can see the clear cardiomyocytes beautifully forming the array, right? Like this. But when we overexpress the mutant, see that there is a disarray. So that proves that the mutation finally affects the cardiomyocytes. And of course, there is a truncation of protein, which I will not go into detail. And when we analyzed both patient and the control, 800 patients and nearly 700 controls, we found homozygous deletion in the patient, not in the control. Of course, in the heterozygous we found in the patient large number, 10%, only 1.7%. But the odds ratio is nearly 7. But look at the p-value, 4 into 10 to the power 14. It's very highly significant. Having that information, we thought that we should also look at how it is distributed in the population. They are all supposed to be normal individuals. These samples were collected for population study. When we looked at the population, we found surprisingly that 4.5% of the Indians carrying the mutation. And we went back to some of the family to find out was there any cardiac death, but they could not explain, but they said that somebody died in the family. They don't know the reason. That explains that, yes, that is existing in the population, 4.5%, and it leads to sudden cardiac death. Although we found in majority of the place, but we didn't find that in Andaman and Nicobar Islands. We know that their origin is different. They came from Africa and settled here. Again, the people from Northeast, they're genetically more closer to Southeast Asian, East Asian. Again, they don't have that. Then we thought that having the data from here, we also should look at population from all over the world. Then with the help of our collaborators, we screened individuals from almost all the continents. And to our surprise, that we found that the yellow ones are the ones which carry the mutation. We found the mutation exists only in South Asia. Apart from India, this was found in Pakistan, Sri Lanka, Malaysia, and Indonesia. And later we found that the Indonesian and Malaysia, those who carry the mutations, have the Indian ancestry. So that means, of course, I didn't tell you that mutation, we sequenced the complete gene, we compared with the chimpanzee sequence, and we estimated that mutation must have arose in India about 33,000 years back. Because it's very old, and the heterozygous condition, it's not lethal immediately, and the individual carry the heterozygous mutation going to die only after the reproductive age. Therefore, it got well spread throughout India, and we have seen that there was admixture between the population, therefore it was well spread within India, and people that there was a migration from India to other places also. So that explains that why did it is there only in um, South Asian population. So that means about 1% of the world population is carrying this mutation. This is just one single mutation which leads to sudden cardiac death. So we don't have that in any other population. And this also gives a clue that why Indians are more prone for heart attack, because we have very unique set of mutation which was not found in other places. This is only one example. Like that, there are so many diseases. You can take any disease, any gene, the Indian genotype is totally different. So that's about the indigenous population. If you look at what the implications of some of the population who recently migrated and admixed with other Indian population. One example is Siddhis. The Siddhis, again, they typically look like African. The reason is they have been brought by Portuguese traders and sold to Nawabs and Sultans to work in their army. Okay, that was about 400 years back. So after they come to India, they started admixing with the Indian population. Therefore, you see that when you have the PCA plot, all these Indians and Yorubans, Africans, and they form a cluster somewhere in between. So they have mixed genome, which I'll tell you in the next slide. So when we looked at different genome like autosome, Y chromosome, and mitochondrial genome, you can see that different genome has these Africans, Indians have different proportions. The highest is the mitochondrial DNA. So that can explain that mostly the males have come because to work in the armies. Then they started marrying local Indians, right? Therefore, they have more of Indian than the Africans. And what is the medical problem here? Is again that's something to do with adaptation to other things. Since malaria is more prevalent in Africa, and most of the individual acquired a mutation in G6PD gene. It's called as A- variant. There are two SNPs, one in exon 4 and the other one is in exon 5. So if the individual has A- variant, they are naturally protected against malaria. About 50 to 60% of the individuals in sub-Saharan region, they have A- variant. But now you see only 10%. Siddhis from Gujarat, Siddhis from Karnataka, they have about 10% of the individual carrying A- variant. That means because of the admixture, their naturally acquired variation which gives protection against malaria has been reduced. But on the other hand, although I don't have data because we are writing the paper, we're not published yet, this is a Jewish population. You know that breast cancer is more common in Jewish population. So they have marker which can, which are susceptible for breast cancer. But because they admixed with the Indian population, that marker frequency is diluted. So they have now advantage of not having breast cancer. And to tell some infectious disease, particularly the malaria among the Indian population, we analyzed few genes, particularly this IL-4 gene, where there's a mutation in this position, plus there's a VNTR repeat. It was interesting to see this the ancestral tribal populations who are in jungle exposed to malarial parasite. Because of the constant exposure of malarial parasite, so they acquired mutation which gives them no resistance against malaria. Leave about the patient and the control. But the population who have been exposed to malaria for a long time, they have now advantage because they have mutation. Similarly, there was another study, recent study, where we found the MBL haplotype, because a low MBL haplotype, MBL gives more protection against malaria. The severe malarial patients has more low MBL genotype compared to the asymptomatic. So again, there's a different kind of genotype is existing here in Indian subcontinent. So we have looked at different aspects, origin of human in India, migration, and the disease susceptibility, and so on. But now, with the genomic information, we know the complexity of Indian population. But without knowing anything, Mark Twain is an American writer. So he has written about India. I read that India is the cradle of the human race, the birthplace of human speech, the mother of history, the grandmother of legend, and the great grandmother of tradition. Our most valuable and most instructive materials in the history of man are treasured up in India only. So I don't know how did he predicted that long ago. Okay. So with this, I'll conclude my talk and would like to thank all our collaborators, including from CCMB, Dr. Singh, and many other students and collaborators from Harvard. David Reach has been a very, very good collaborators, Chris, and many others. The list goes. Otherwise, without their support, it would not have been possible to do such a wide study. And thanks to the funding agencies, UKRI, CSIR, and ICMR. Thank you very much.